So let's take a look at the eukaryotic cell cycle. So there are two main phases. We have interphase and the M phase. So during interphase, the cell is going to be growing and doing its jobs, whatever it is, and most of the cell's life is going to be spent during interphase. And the cell is not dividing at all when it's in interphase. And there's three phases in interphase. There's the G1 phase, S phase, and the G2 phase. And then during M phase, the cell is going to be dividing. And M stands for mitosis. And mitosis is when the nucleus is going to divide. And that just means that we're going to be dividing up the chromosomes and splitting them into the two different daughter cells. And then cytokinesis is when the cytoplasm gets divided into the two different daughter cells. And then that's when the two daughter cells will separate and become their own cells. So let's just take a look at a picture of the cell cycle of a eukaryotic cell. So here's when a new cell is going to be formed, and it will enter interphase. Interphase is this whole time frame here in gray. And so you can see interphase consists of the G1 phase, the S phase, and then the G2 phase. And then here's the M phase. So you can see the M phase is pretty short, and this is when the cell is dividing. And so it consists of mitosis and then also cytokinesis. All right, so during interphase, the cell is just growing, doing its normal jobs, and it's just getting ready for mitosis. And so we've got three phases during interphase. The first gap phase is our G1 phase. S phase, that's when the chromosomes are going to be duplicated or replicated, and S stands for synthesis. So synthesis just means to make, so you're just going to make a copy of the chromosomes because you need to give one copy of the chromosomes to one of the daughter cells and the other copy to the other daughter cell. And then the last phase will be the G2 or second gap phase before we enter the M phase. Okay, so here's where we are, first gap phase. Here's our S phase where we're going to copy the chromosomes, we'll duplicate or replicate them. And then here's our G2 phase before we enter into the M phase, and then the cell will divide. Okay, so important to remember during the S phase of interphase, so just remember S means synthesis. So this is when we're going to make a copy or duplicate or replicate the chromosomes because we need to make sure that we've got one copy of the chromosomes to go to one of the daughter cells, and then the other copy of the chromosomes will go to the other daughter cell. And so when we copy or replicate these chromosomes, we're going to end up with these two sister chromatids. And these two sister chromatids are going to be joined together in the middle here at what's called the centromere. And so this would be one sister chromatid, and then the other sister chromatid would be over here on the other side. And these sister chromatids are identical to each other because they're just a copy of each other. And so one cell is going to get one copy of this sister chromatid, and the other cell will get the other copy of the sister chromatid. And once they're separated, each one will become a chromosome again. Okay, so here's just a picture just showing you've got your original chromosome, and when the cell goes through S phase of interphase, we'll make a copy of that chromosome. And so we just join together those two copies, those two sister chromatids at the centromere here, and that is your copied chromosome there during S phase. Okay, so once we hit the M phase of the cell cycle, we're going to have the cell divide. So it's going to do mitosis and then cytokinesis. So in mitosis, we're going to separate the two sister chromatids into two chromosomes, and one copy of the chromosomes will go to one daughter cell and the other copy of the chromosomes will go to the other daughter cell. So each of the daughter cell will have a copy of the chromosomes. So they'll be genetically identical to the parent cell and also genetically identical to each other. And mitosis is going to be divided into 5 stages, which we'll talk about. We've got prophase, prometaphase, metaphase, anaphase, and telophase. And then cytokinesis is when we're going to divide the cytoplasm between the two daughter cells, and then the two daughter cells will finally be separated from each other and become their own cells. Okay, so here's just a kind of a simplified overview of mitosis. We've got our parent cell that we're going to start with here. We've got our single chromosome. And then here's during S phase of interphase, where we're going to make a copy of that chromosome. And these two sister chromatids are going to be joined together at the centromere. And then during mitosis, we're going to separate those sister chromatids into the two daughter cells. So each daughter cell gets a copy of that chromosome. And so now the sister chromatids now are called chromosomes again. And you can see that these two daughter cells are genetically identical to each other. And they're also genetically identical to the parent cell that we started with. So during mitosis, we're just making clones of the parent cell that we started with. Okay, so here's our cell cycle picture. Again, we've got interphase, G1, S phase, and G2 phases. And then here's our M phase, and we've just kind of listed out the different phases during mitosis that the cell goes through. And then here's cytokinesis. And then you can see at the end, we're going to produce two genetically identical cells. Okay, and then here are all the phases of mitosis and also cytokinesis. So here's G2 of interphase right before we go into the M phase. And then here's our first phase of mitosis. So we go through prophase and then prometaphase, metaphase, anaphase, and then telophase and cytokinesis kind of occur together here. And then in cytokinesis, we're going to divide the cytoplasm between the cells and then finally separate these two cells so then they become individual cells. And these two daughter cells will be genetically identical to each other and to the parent cell that we started with.